Hello, my name is Flanagan, and I am a senior at Beloit Memorial High School. This semester, I am a welcome intern here at the Welty Environmental Center. Thank you for taking interest in my project. My GEOPAths project is on the soil particle size distribution in the prairie and forest habitats here at Big Hill Park. To begin this project, I did some background research on soil particle size distribution and how it may vary across different habitats. First off, soil particle size distribution is the different amounts of soil particles within a sample. The three main particles are silt, sand, and clay. Forest samples have been found to have a makeup of majority sand and silt with little amounts of clay, while prairie samples are also mainly sand and silt. They have higher percentages of clay within the soil than the forest would have. These soil makeups are important to what types of plants grow in a habitat, along with what animals and organisms inhabit these areas. So, this leads to my main research question. How does the prairie and forest soil particle size distribution compare here at Big Hill Park in Beloit, Wisconsin? I started collecting soil samples and conducted testing from early April to May 10th, 2022. In each habitat, I sampled 3 sites in each habitat type. I followed methods according to the globe pedosphere protocols. To collect the samples, I used the auger method to retract the soil from the ground and laid it on a tarp. I then identified the different soil horizons by using Globe's soil color book and identified the texture of the soil through the process of ribboning. Along with that, I also tested for carbohydrate levels by spraying a sample with vinegar and recorded whether the soil bubbled or not. As for the main soil testing, I started with mixing 25 grams of sieved soil with 50 milliliters of detergent solution, and then you let that sit for 24 hours. Then I filled the 500 milliliter beaker with the solution and the remaining amount you needed to fill the beaker with water. Next, you mix the solution by turning it hand over hand 10 times. And lastly, I tested the soil samples through 24 and 48-hour hydrometer and temperature readings. As for the results, the prairie samples were mostly sand and silt. However, the clay percentages in said samples were higher than the clay levels in the forest. In the prairie samples, the clay percentage stayed constant throughout the horizons. However, within the forest samples, the clay percentage increased as the depth increased. In both habitats, the levels of the silt increased as the depth increased. As you can see in the graphs, it's pretty interesting to see the percentages change as the depth increases. In conclusion, the forest samples came out to be a loam soil, which is ideal for tree growth, as well as small shrubs and any other undergrowth plants in the forest. As for the prairie samples, they were commonly a sandy clay loam, which is ideal for switchgrass, big blue stem, Indian grass, and many other different types of grasses that grow in the prairie. So it's super interesting in the graphs to see the soil's characteristics, which helps us identify, what plants grow in those habitats, what organisms use those habitats. So it's interesting to have the graphs as a visual to show that soil samples make up. I would like to thank the National Science Foundation and the UW-Madison Center for Climatic Research at Nelson Institute, along with Aaron Wilson and Michael Notaro for logistics support, and finally, Darien Becker and Bennett Donovan for field and project support.